let's walk through a couple practice questions about complementary base pairing. Now, as the video goes along and the question is asked, you should be prepared to stop the video so that you can try to answer the question on your own. And then when you come back, we'll talk about it together and I'll explain how you can potentially try to address the question so that you can come to the correct answer. Okay, here is question 1. You're a scientist and have determined that a sample of DNA you have contains 20% adenine. So the question is, what percentage of guanine does your sample contain? Now stop the video here because you're going to have to do a little bit of math and think about it, and then come back and we'll talk about it together. Okay, did you stop the video? I hope so. So how do we address this? What information do we have? Well, we know it's DNA, so we know it's double-stranded. We also know that the sample we figured it out, that it contains 20% adenine. So at this point, we have to think about complementary base pairing. And we know the DNA is double-stranded. And if there is an adenine on one strand as the base of this nucleotide, then the nucleotide on the other strand is going to have a thymine. Similarly, we know that cytosine and guanine have a complementary base pair with each other. So, if we have, for every adenine that we have in DNA, we know there's going to be another thymine for that adenine because they base pair together. So it's kind of like if you think of your shoes, if you have like 10 pairs of shoes and you have the left shoe for 10 pairs, then you also have 10 right shoes for those pairs, right? So if you have 10 adenine bases, then you're going to have 10 thymine bases in that DNA, and it works the same way with percentages. So thinking about that, we know then that the percentage of adenine in our DNA sample is going to be equal to the percentage of thymine in our DNA sample. Now, in the question, it gave us that there's 20% adenine in our DNA sample, and thus we know there's 20% thymine. Because remember, for every adenine, there's going to be a thymine that's making a complementary base pair with it. Okay, so now we know that adenine and thymine together are going to be 20% plus 20%. So 40% of our DNA sample is adenine or thymine, A or T. So thinking about it, we know in DNA, there's only two other bases that exist, cytosine and guanine. So the remainder is going to be guanine and cytosine together. So if we have 40% adenine and thymine, that means that, 100% minus 40% is 60%. 60% of the DNA is guanine and cytosine together. Now, the question wasn't asking for them together. The question was saying what percentage of guanine does our sample contain? And if we know that guanine and cytosine together are 60%, well, look at the graphic. We also know that for every cytosine, there's going to be a guanine. So that 60% is half guanine and half cytosine. And so to figure out the rest, we know that, if guanine and cytosine together are 60%, half of that is going to be guanine. So we're going to have 30% guanine and 30% cytosine. And there's our answer. Our DNA sample contains 30% guanine. And at this point, it's good just to check your math to make sure that it adds up to 100%. We know that adenine, thymine, that's 20% each, so up to 40%, and then 30% guanine and 30% cytosine, so 40% and then 70% and then 100%. We're at 100%. Okay, here is a very similar question, so pause the video and see if you can answer it now that we've discussed the first one. This question says we're a scientist, we have another DNA sample that contains 40% cytosine. All right, so our question is, what percentage of thymine does our sample contain? And it's the same reasoning as before, right? Except we're starting with knowing about cytosine. So we know that for every cytosine in DNA, there's going to be a guanine that it base pairs with. So the percentage of cytosine in our sample is going to be the same percentage as guanine in our sample. So if we have 40% cytosine, that means that we're going to have 40% guanine. Now, what is the question asking for? The question wants to know what percentage of thymine does our sample contain? Okay, well, we know there's cytosine, guanine, thymine, and adenine. So the other two are adenine and thymine together. Cytosine and guanine are 40 and 40. So those two together are 80%. That means that adenine and thymine together are going to be 20% of what's left. Now remember, these are both of these together. So that means you have to divide this in two, because half of that 20% is adenine and half is thymine. So that means that 10% of our sample is going to have adenine and 10% of our sample is going to have thymine. And then again, let's just do the math just to make sure that adds up to 100. So 40% cytosine, 40% guanine, 10% adenine, and 10% thymine. That equals 100%. So we're good to go. So to answer these questions, look at what you have. Think about which base that particular base does a base pair with, and that way you can figure out what proportion of the total DNA is that particular base that you're given, and you can go from there to figure out the base that they're asking for. Okay, here's another practice question about complementary base pairing. Determine the complementary strand to the DNA sequence you have here. Pause the video and we'll chat about it when you come back. Okay, so in this question, we're looking for the complementary strand. And complementary strand means that, well, this is a piece of DNA. We know this is DNA because we see it has G, C, A, and T. If it were a piece of RNA, we wouldn't see the T's, right? We wouldn't see thymines. Instead, RNA has uracil. So just by looking at this, we assume it's a piece of DNA. And we need to know the complementary strand. This question is just asking, okay, what is the complementary set of base pairs that correspond to what we see here? So again, let's think about our complementary base pairing rules. We know that adenine is going to pair with thymine always, and that cytosine is going to pair with guanine always. So if we come up here, we see here's guanine, guanine, cytosine, adenine, thymine, thymine, guanine, cytosine, adenine, cytosine, cytosine, guanine, thymine. I hope I said that correctly. And now we just have to look and say, okay, well, guanine always forms a base pair with, here it is, cytosine. So the complementary strand would have a C there. Right, here's the next one is also a G, so there'd be another C in the second position here. The third nucleotide we see here has a cytosine base, so the complementary strand would have a G, a guanine, and it goes on from there. So the answer to this puzzle, this question, would be CCGTAA, CGTGGCA. So for every guanine we see in the original strand, there's going to be a cytosine in the complementary strand, and the opposite is true as well. For every cytosine in the strand given, we're going to have a guanine in the complementary strand, and then the same is going to be with A pairing with T and T pairing with A. So there we go. This is the complementary strand. Now this is question 4. It's a very similar question, except now we've been given the ends of the piece of DNA. And we know that the ends just indicate to us the forward direction of DNA. So the forward direction is always from the 5' end to the 3' end. That's the forward direction. So what we're looking at here is sort of exactly the same. The first thing we have to do is find the complementary sequence of nucleotides. So pause the video and see if you can quickly figure out what the complementary strand would be as far as the sequence of nucleotide bases. So to figure out question 4, we address it just like we did with the previous question. We think about complementary base pairing rules, which is that thymine and adenine are going to form a base pair together, and that guanine and cytosine on different strands are going to form a base pair together. So when we're thinking about this, we know that if there's a thymine here in the strand that's given us, then we know there's going to be an adenine in the other complementary strand, right? So by doing this, we can figure out what the bases are in both strands of our DNA because we know the DNA is double-stranded. And here we have the sequence of nucleotides that's found in both of those strands. The one thing that's a little different here is this question gave us, the 5' and 3' ends. So now we have to figure out, okay, well, we probably should label our new strand then as well, for full credit. So if that top strand is 5' to 3', right to left, then we know that the bottom strand is going to be in the opposite direction, 5' to 3', because DNA is antiparallel. One strand of DNA goes in one direction, and the other strand of the DNA goes forward in the opposite direction. So if our top strand is 5' to 3' right to left, then our bottom strand is going to be 5' to 3' left to right. So a way to double check yourself here is you should never have a 3' end and another 3' end on the same side. Okay, so with these two strands, you'll never have two 3' ends on the left or two 3' ends on the right. They have to be opposite because they're going in different directions, and that's what the 5' to 3' indicates to us. The beginning of the strand and the end of the strand. Okay, so we started with a strand here that was 3' to 5'. We can still determine the complementary strand, right? We just have to label our ends so we know in which direction they're going. Now here in question 5, this is a slightly different question. Instead of the 3' end being on the left, it's the 5' end, and it's asking us to determine the complementary strand. So pause the video and see if you can write out what the sequence would be on the complementary strand of DNA for this one shown here. So to address question 5, we're going to use the same complementary base pairing rules we have before, that if there's an A or an adenine in this strand that's been given to us, then we know in the complementary strand there's going to be a thymine, right? So this sequence would be TAG, CGA, ACTT, GC. That is the complementary sequence to what's been given to us. Now the next thing, of course, is to figure out the different ends. And if the 5' end is on the left side for the strand that they gave us, then we know in our new strand, the 5' end is going to be on the right side, correct? Because we know that they go in opposite directions. This time, the strand that they gave us for this question was going left to right. In question 4, the strand was going right to left, 5' to 3', but now it's 5' to 3', left to right. And that means that in the strand that we've determined, we know the 5' is going to be on the right because it needs to go in the opposite direction as the strand that they gave us because we're dealing with DNA here. Now, the next follow-up question to this is to write your answer in the forward direction, which is 5' to 3'. And here, just to, so we can keep everything straight, this was our answer. So pause the video and see if you can rewrite our answer in the 5' to 3' direction. So for question 6, we have to think carefully. What are they asking us to do? Are they asking us to determine the complementary strand? No. So we don't have to worry about the complementary base pairing rules of thymine base pairing with adenine and cytosine base pairing with guanine. This question is simply asking us to write the answer in the forward direction. And we know the forward direction is 5' to 3'. Now in this case, it's written backwards then, right? Because the 5' end is on the right. So if we were writing this in the forward direction, we would just rewrite it. Now let me give you a real world example of this, okay? So here I've come up with a random arrangement of letters, but it's actually going 5' to 3' right to left. So this is actually written backwards. If we want to write our answer in the forward direction, instead of having it written backwards, we just rewrite it starting with the 5' end now on the left. So here we go. I'm going to rewrite it with the 5', the first nucleotide over on the 5' end, that's a T. So it's going to be T, H, A, T, C, A, T. See what I'm doing? I'm just writing it backwards now. Essentially, I'm making it so that instead of being written backwards, I'm rewriting it so it's written in the forward direction. So it's kind of like the one up top is sort of the mirror image of what you need to write out. And now the 5' end is on the left. So now we wrote it in the forward direction, 5' to 3'. And just so happens that with this particular example, if you write it in the forward direction, you see that it says that cat is huge. This is an homage to my cat Lucky, who is indeed a huge cat. So when we're thinking about this, we need to see what the question is asking. Is it asking us to determine the complementary sequence, the other strand of DNA? Okay, that's one thing. That's what we did in question 5. But question 6 is really just asking us to write it in the forward direction. And I know you might be asking, like, why? Why can't we just leave it with 3' there? Well, you know, it's convention to write it in the forward direction. Like when you write words on a screen, you write them in the forward direction. You don't write everything starting at the end. So it's convention in biology to often write a strand of DNA in the forward direction, starting with the 5' nucleotide and going all the way to the last nucleotide, which is at the 3' end. The other thing is that, you know, sometimes you'll need to maybe write it in the 5' to 3' direction so you can do something else and manipulate it in a different way. Now, a lot of, you know, questions and professors might not ask you to do this, and maybe they'll set it up for you so you never have to rewrite it 5' to 3'. It's, you know, maybe your answer will always be in the forward direction when you actually write it out properly. But you know, it's good to know and understand the different sides so this way you can work with it and manipulate it and always double check the ends because you don't want to fall victim to that trick question where they ask you to write it in the forward direction and you have to just rewrite it, you know, so that the 5' nucleotide is the first one and the 3' nucleotide is the last one. Okay, so being that this strand of DNA is backwards, it's starting on the right side and it's being written, you know, it's written backwards, we just have to essentially rewrite it in the forward direction so that that first nucleotide, which on the 5' end, over here it's C, G, T, T, C, A, G, C, G, A, T. Right? And you can see here it is. I just wrote it out. Here's the 5', C, G, T, T, C, A, G, C, G, A, T. This wasn't asking for the complementary strand, right? This isn't asking us to use the base pairing rules to figure out the other strand. In fact, that's what was given us up here. So if we used this and we found the complementary strand to it, it would have been this one that was given to us at the beginning. It's really just asking us to write it in the forward direction. And here it is. Now we're writing it from 5' to 3'. So for questions like this, you need to ask yourself, what am I being asked to do? What is the question asking me to do? Is it asking me to find the complementary strand sequence? If that's the case, then you need to use your base pairing rules that adenine always in one strand always forms a base pair with thymine in the other, and the same is true with cytosine in one strand and guanine in the other strand. Or perhaps the question is asking you to write a DNA sequence in the forward direction, and that would be the question 6 that we just did, right? Maybe it was written backwards, so we had to turn it around so that the 5' nucleotide was first, and then we rewrite it until the 3' nucleotide is the last one. Or perhaps another question might ask us to transcribe this DNA into messenger RNA. Today, we only figured out the the complementary strand of DNA. So we replicated, this piece of DNA, but a similar process occurs if we're creating messenger RNA from a DNA template. So there could be a question where they're asking you to create messenger RNA from that DNA template that they give you. And that's a process of transcription, right? Transcription is creating a piece of messenger RNA from a DNA template. And you can check out another one of my videos if you want some practice questions about transcribing DNA into messenger RNA. Now, one last question. Determine the complementary strand and write your answer in the forward direction. Now, I've already figured this out for you. Here was the strand you were given up at top, and the complementary strand right here is at the bottom. We've already put in the ends. We know that here's the 5' end of the strand we were given. So this piece goes from right to left. That's the forward direction. So we know our new piece, when we're putting the ends down, is going to go left to right. 5' to 3' is the forward direction. Because again, DNA is anti-parallel, that means that the two strands are parallel, but they're going in opposite directions. Just like a road, right? The two sides of a road. So this question says, with your answer, write it in the forward direction. And here's our answer down here in gray. But this is kind of a trick question. In fact, it is a trick question because our answer is already written in the forward direction. It is written 5' to 3'. So this is a trick question asking us to essentially write your answer in the forward direction. Well, it's already written in the forward direction because the template that they gave us to work off of was going right to left in the forward direction. So our answer on the bottom is going to go left to right, 5' to 3'. Maybe this wasn't a trick question. Maybe it was just worded badly. Maybe they just wanted us to, mark out which side was the 5' and which side was the 3' end, which we did. And so that reveals to us that our answer is already in the 5' to 3' direction.